So I think we can start then. Richard, are you in good shape?
Yes, give me a moment.
Yeah, so Adam, if you want to make, because you'll be the host of the meeting now, so if you want to make Richard the host, and then he'll be able to start sharing his screen.
Sure, will do.
Okay, great. So first, while that's being done, I'll say hello to everybody joining from around the world. Richard obviously is in the US. There's plenty from the UK. I'm currently in Asia, just visiting some colleagues. So it's great to have you all here. So this is the Tissue Image Analytics Center seminar series. So we did quite a few seminars last year, last academic year. We've taken a break over the summer, so it's good to be back. So this is the first seminar in the autumn semester, 2022 to 2023. So these seminars invite researchers, mainly from the area of computational pathology, to give a seminar on their recent work in the area. So we try and treat this as an interactive seminar. So if you have any questions, please just raise your hand or type in the chat and we'll field those as we go. So today we have Richard Chen. So it's great to have you here, Richard. We've been following your work. So Richard is a fourth-year PhD student where he did his bachelor's and master's in Johns Hopkins University in the US, specializing in biomedical engineering. And since then, he's now a PhD student at the Mahmoud Lab at Harvard Medical School in the US, where he's specializing in developing solutions for computational pathology, for example, interpretable AI solutions, and also multimodal solutions, specifically looking into combining computational pathology and genomics. So I think with that, hopefully you're now the host, Richard, and
yes,
we can hand it over to you and you can present when you're ready.
Oh yeah. So also thank you for the great introduction, Simon. Like Simon said, feel free to, if you guys have any questions, curiosity about a slide, just feel free to just stop me and just ask more. Okay, so yeah, so kind of today's talk for the TIA seminar series is my work on like exploring context-aware and multimodal computational pathology. So this is a culmination of all the work I've been doing since 2019. There's been some evolution in like kind of the work that happened in 2019 versus the work that's like very recently happened. So yeah, so I'm very excited to share with you this like journey. Yeah. So kind of the problem that I've been like kind of working on throughout my PhD is thinking about weakly supervised learning for these gigapixel pathology images. So unlike natural images, you know, whole slide imaging is a very challenging computer vision domain in which these image resolutions can be around 150K, 150K by 150K pixels. I'm going to play this animation right here. Yep, 150K by 150K pixels. So the right-hand side, you know, is coarse view of the whole slide from which we can magnify and zoom into fine-grained details such as tumor and immune cell co-localization, which are, you know, for certain tasks are prognostic, such as, you know, cancer survival outcome prediction. Though weakly supervised AI can potentially be developed for cancer prognosis using whole slide imaging in the broader context of current clinical management of cancers, cancer prognostication is a multimodal problem that's driven by markers in both histology and genomics and other clinical data such as age and potential age and gender. So we not only want to look at potentially, looking at how to use pathology alone for cancer prognosis, but also incorporate all of these other modalities into kind of the supervised learning problem. This is just like a brief rundown of kind of the current paradigm, or one of the current paradigms for weak supervision, which roughly goes as follows, where we have this whole slide, we formulate it as a set of non-overlapping patches, typically of size 256 by 256 at 20x, but a lot of other works have explored 512 by 512 at 20x or size 256 at 10x. So at this magnification, we kind of patch, do feature extraction, and then pooling to compute a slide level feature representation Some works, of course, pre-extract all the feature embeddings and then do the pooling. But more recently, there have been some works doing end-to-end. But overall, this is like kind of like the rough architecture of how weak supervision follows in pathology. This method works well for a lot of kind of classification tasks with easily discernible visual concepts. For example, if you're doing subtyping of let's say if you're doing subtyping of a certain disease type, such as let's say clear cell versus papillary, you know that the histological signatures for a clear cell won't appear in papillary and vice versa. So very great for kind of very easily discernible visual concepts, but I know survival prediction is a much more difficult task that entails capturing important contextual and hierarchical information in the tumor microenvironment, and it's limited by in this kind of previous framework. Limited small patch sizes have limited context and also create very large sequence inputs. From my estimation, if you're patching at 20x with 256 size patches, you roughly create 15,000 patches. And this is a super large sequence length that we're feeding into our model. And that results in kind of MIL approaches being limited to only permutation and variant pooling. So because of how large number of instances are, kind of most methods are restricted to mostly kind of these permutation and variant pooling methods that may not be able to capture context. And I think lastly, I just want to talk about how, even if we can equip, these weakly supervised models with more complex aggregation schemes, slide tasks are high dimensional, low sample size problems, and thus more complicated MIL architectures may lead to over or underfitting. And of course, this is both, we also ideally would like this to be multimodal and also interpretable for clinical translation. This is just to, this is also, lastly, so on the interpretability, in developing prognostication tools for tangible clinical translation, the goal for deep learning based survival outcome prediction is to not only perform better patient stratification, but also using these models for potential biomarker discovery, specifically using model interpretability to elucidate novel morphological features not captured in current clinical readouts. This is a great, one of the many great reviews for computational pathology, but I'm really like this figure because it kind of isolates both like kind of the basic CPath tasks that everyone's doing well on, and then the advanced CPath tasks that I think there's been some recent successes, but more ways to go. And I think the main difference that this figure is kind of isolating is that these advanced CPath tasks, the labels are kind of more more objective, like like overall survival response to treatment and or mutation prediction. So these are tasks that pathologists can't do. Pathologists can't can subtype, they can grade, but they can't infer more things such as, will this patient respond to a treatment? So kind of like the dream is that if we train these deep learning models well, maybe if we use interpretability, they can they can review novel information. So that's the brief introduction I'll give. I'll jump into kind of like the works I've pursued that have kind of addressed different aspects of these problems. And with the first work, starting with the most recent work in cancer cell. So kind of just to go over some of like the more clinical motivation again, so the kind of the current standard of care for cancer prognosis is this like subjective and qualitative analysis of histology images, which can suffer from large intra-observer variability amongst pathologists and many cancers. So there is some unexplained variance as to why some cancers will recur, or why patients with similar pathology and or grades will have different outcomes. Also, over the course of the years, I think the taxonomy for grading has also gone on to include. does genomic information come from the same block as the whole slide? Yes, it would. Yes, it would. So kind of, in recently with the advent of next generation sequencing, there's been a lot of interest in also using genomics and transcriptomics for also doing patient stratification. So many cancers are currently undergoing revision of like kind of the cancer taxonomy. So for example, for diffuse gliomas in 2015, it was recently revised to kind of stratify patients by both histo and molecular signatures, such as oligodendroglioma, IDH mutant astrocytoma, and IDH wild type astrocytoma. So cancer prognosis is, like in the current clinical management, a multimodal problem. However, the assessment of histology is still manual. One of the at the time of this work, this was around 2019, 2020, kind of the mainstay approaches in using machine learning for survival prediction has been kind of mostly unimodal deep learning networks, where, for example, in for images, Mobadersani et al. Showed that you can use CNNs for supervised with a Cox loss to stratify outcomes, and similarly in Yousefi et al., all from Lee Cooper's group, they also showed that you can supervise feed forward networks on top of genomics, also with Cox loss, and these methods do quite well in terms of patient stratification, but overall these methods are not integrative. So this is kind of an overview of the Porpoise work. So what we did was, okay, let's for molecular data, we use both genomics, mutation, copy number variation, and RNA-seq data, and we're like, okay, we're gonna for processing genomic data, we're just gonna use feed for layers. For on the whole slide imaging side, we use attention multiple instance-based learning for the canonical approach for kind of pooling all of these for kind of patching all of the tissue regions in the whole slide, and then doing feature extraction and then pooling. And then, however, to kind of combine everything together, we use multimodal fusion. So this is based on fusion via the Kronecker product. So we have both of the views of both the whole slide and the genomics, and we're going to combine them together to see if we can do better patient stratification. And on top of that, the objective of this work was to not only do better patient stratification, but also see, can we elucidate, investigate whether multimodal interpretability will elucidate new what kind of information would it find. So this is interpretability on the genomic side, which we conducted via integrated gradients, and also for the histology side, we looked at the high tension regions in the histology image, as well as within the high tension regions, what cell populations and admixtures are used as prognostic information for driving risk.
Richard, did you ever have any issues with missing data, or did you have the genomics, and the other genomic information for every tissue sample that you had?
Yeah. Oh, actually, this would go right into the next slide. So okay. This was the overall study design. It was both unimodal and multimodal comparisons on 14 cancer types with kind of the same splits, the same samples across each split. In terms of like dealing with missing data, we didn't do any imputation. What we did was we kind of looked at the cohorts where there was I think the overall criteria was we wanted to pick cohorts where with with a copy number variation, mutation, RNA-seq, and whole slide, that the overlap would be greater than 150 samples. There were some samples that, if we used everything, it would lead to too much of a dropout. So for example, for certain diseases like endometrial carcinoma, we didn't include RNA-seq because here, going from copy number variation mutation to everything was lost around 400 samples. This was using, I think, one of the more recent data dumps from GDC. So there could be some information that I could not find for endometrial carcinoma, but roughly this was the strategy.
Cool. So you assume that every form of data had to be available for all modalities?
Yes.
Okay.
Yes. But there's a lot of imputation strategies that I will be happy to send you some more literature on that topic.
So I wonder whether you could use, for example, like a multitask approach where you don't assume for the each task to be, you don't need to have each task to be across all of the other tasks, and then you could potentially utilize as much data as possible.
Yeah, I think another strategy that on top of multitask learning, another strategy I've seen pop up is using kind of using generative models such as VAEs to kind of also impute some of the missing data. I think no one's really framed it this way, but imagine you can use something similar to not like a cycle-GANT architecture, but you can adapt from one modality to the other to do computer imputation of guesstimating any missing data. I think for this study we considered imputation. I think imputation is quite important because for example for certain cancer types, including everything, would just lead to too few patients for survival analysis. But I think it was a bit beyond the current what we first wanted to do in this study design.
Great, thank you.
Okay. This was the overall. I'll just jump to some of the overall results. Kind of what we found is that when we train our multimodal model with the comparable unimodal models of the same architecture, multimodal fusion improves on 12 out of the 14 cancer types, and we see statistical significance of multimodal fusion in 5 of the cancer types. So these were how we defined statistical significance was we used a log rank test to kind of see whether or not the model could stratify the top 50 and lower 50 percentile patients. And so then these were cohorts where just using molecular data alone could not do patient stratification, whereas multifusion was successful. And there was a few cancer types where this did not improve. For example, a lot of the renal cell carcinomas and endometrial carcinoma, where unimodal models do better, if not more comparable. But at the same time, I think one of the things that we wanted to see was, is multimodal fusion necessary in all scenarios? So some of these results suggest that we would not need multimodal models in all settings for doing cancer prognostication. So this was more of the quantitative results. I want to jump into more interactive session where this was kind of one of the main tools that we've developed called Porpoise. So for pathology omic research platform for integrated survival estimation.
Sorry,
this, name took yes.
Hey, before sorry, I have a question. Yeah, before you going into the interesting demo. So I wonder if there is So when you tested with only omic features, right? With only omics.
Yeah.
I wonder if the omic features which proved to be important is in that model, but also picked up in multimodal, or no, it was different.
I think they tended to be different. I guess I'll go into, actually, that's probably a very interesting area of interest that I want to go into next. Maybe to answer that question. Okay, I'll answer your question in maybe like 3 minutes. But let me, but I'll first show kind of the overview of this, and then I'll talk about how what I think is like I think there's some like problems in like training multimodal networks, and I think there's like some dynamics in integrating pathology and genomics that haven't been elucidated yet, and I'll explain like the future direction of that line of work. Okay, so this is kind of, so overall we have, so this kind of platform has 14 cancer types. We has a lot of it has patients from across from all the different validation folds, and this is just showing kind of the interpretability, local interpretability. So these are the interpretability for an individual patient, and then the global interpretability. So this is interpretability across the entire cancer cohort. We have this both on the histology and the genomic side. So you can go in kind of the red are regions that are prognostic, so used by the model for doing survival analysis. And then you can see how interpretability would shift when we're going from unimodal to multimodal. So here, just like more of a coarse view, you can kind of see how attention shifts from maybe, let's say, from let's say this kind of region over here, which looks like, I'm not a pathologist, but this almost looks like, yeah. So but we can see how interpretability shifts from going from unimodal to multimodal. And lastly, and then also we can view the same thing. We can also see a lot of the molecular features used for locally for driving risk in this patient. And then so we can see both the and also these are the overall features used across the entire cancer type. Maybe just to show a more canonical cancer type like glioma here, we can kind of see kind of the more canonical like molecular biomarkers such as IDH1, ATRX, and then TP53. We can see this for both overall mutation, CNV, and RNA. So this is kind of and I think hopefully this is just like a kind of a more resource platform to kind of help pathologists and researchers, kind of see what multimodal models are learning in survival analysis.
That's super interesting. Do you know when you toggle on the multimodal, I don't know whether you've had a chance to discuss with pathologists whether the attention then makes more sense? Like, is there a way of explaining why that shift is happening?
Actually, so I want to just jump. I want to jump towards one. Maybe if I can find it. So there are a couple of cases that I found where the heat maps just completely inverted. So here it looks like it's mainly focusing on like stroma. Just jump towards the end. Oh, there are some, I guess, slides that we need to fix. Let me just look at one more.
On in the breast cancer, you can go first sample.
I think one of the main weird things that we found was that when we used multimodal networks, some of the interpretability heat maps tended to just shift. So whatever kind of the unimodal model looked at, the multimodal model just looked at the complete opposite.
Okay, that's interesting.
Let me find an example of like a big shift right here. See if we can find it.
You got a lot of slides to look through here.
I got a lot of sides here. I'll just jump maybe like the first one. I think the first one was like a good example. But here you can kind of see that this is like clear cell RCC. So the unimodal model was looking at kind of this chicken wire vascular pattern. However, when we go from unimodal to multimodal, the interpretability shifts away and looks more broadly at the stroma. I think one of our hypothesis on why this happens, I think this is still something that we're still trying to figure out. But why this happens, I think, is that when you're using molecular information, like the genomics is almost like a shortcut that is almost like a pattern shortcut where if you're giving it tumor information, let's say if for let's say survival prediction in renal cell carcinoma, if you're giving it the mutation status of like BAP1 and in VHL, then kind of the model already knows that that tumor signature is present. So, and then because that tumor information is made available on the genomic side for when it's trying to find prognostic features in the histology, it doesn't need to kind of attend to where the tumor is, because that information is already given on the genomic side. So in a way, I think I kind of view like genomic information as almost being like a shortcut in like multimodal learning. And I think this is a fact. I think there are some, interesting, I think kind of like tangential to like all the works in like multimodal gene mutation predictions, like, like all the works that, that your group and Cather have been doing. I think where we're trying to predict the mutations status from one modality. I think there's some shared mutual information between both modalities that, and I think this mutual information may affect multimodal training. And then there's also modality specific factors in which you can't predict certain mutations from histology. So this is where kind of for multimodal learning, it would you would want to doing interpretability on those cancer types would doing interpretability on those cancer types, you'll find very different kinds of information. This is all kind of my hypothesis on like what's going on, but I think it's a very cool thing to see. So
it's a very interesting hypothesis. We've got 2 questions to field, if that's okay, Richard. So first of all, from Clara, she asks, what would your explanation be that multimodal does not always improve C-index performance for some cancer types?
I think for why multimodal would not always improve, I think my hypothesis would be that there's a lot of shared mutual information between both modalities. So as a result, there's not any prognostic information given by one modality that would kind of that's not captured in the other modality.
Okay, perfect. And then we'll go on to Mateo's question. We've got quite a few questions coming in now. I'll save some for the end, but let's go to Mateo's question and then we'll carry on.
Yeah.
Have you tried to use also spatially resolved genome expression? Maybe that could help in understanding the disagreement.
Regarding that, it's actually kind of where I wanted to kind of go, was like kind of explore some of the early fusion work for like doing multimodal fusion, but I think to include spatial transcriptomics and other spatially resolved technologies, it can be very expensive. It doesn't have the clinical penetrance yet, but I think that's exactly where kind of this future direction, kind of where the future of like multimodal learning is going.
Fantastic. So Mohsen and Alex, we've banked your questions. We'll ask those at the end. So Richard, please continue.
Okay. I have around 15 minutes left, right? Is that true?
Yeah, you can have about 20 minutes or so.
Okay.
We can really go, because we're asking questions as we go. We can really go up to 0:00, so you've got a bit of time.
Okay. Okay. Sorry, I'll speed things up. I'll speed things up a bit. So this is just for one of this is kind of one of the so for each cancer. So because in the study we kind of have these like cancer vignettes for each cancer type. So this is one I thought was very interesting. I think it's like a cancer type that we're thinking about exploring further. But for pancreatic adenocarcinoma, we actually, so this is an example of a cancer type where, you know, it has poor separation of high and predicted high and low risk patients in molecular and histology alone. But when we combine these both together, we see better separation of survival curves in the low surviving patients. You know, again, this is looking at, you know, using tools such as HoverNet. We can introspect the cell populations of the high tension regions, for which we kind of see more lymphocytes in predicted low risk patients of the high tension of predicted low risk patients, and we see more stroma in the predicted high tension regions in the predicted high risk patients. And then this is looking at kind of how the interpretability that doesn't only shift on the histology side, but also on the genomic side. So here we actually see that, I think this is we actually see that, you know, going from genomics only to then multimodal fusion, we actually see that a lot of there's like lower attribution given to molecular features. So again, what that means is that the importance of molecular features, or at least the impact of these individual gene features for risk prediction, is kind of lowered and reduced. So kind of these features aren't as prognostic when we combine it with histology. So the attribution is given towards more histology than genomics. And thus, not only do we see like different ordering of important genes, but also the relative impact that these genes have is also reduced in risk prediction. And then kind of the main thing that I think our work found that I think was quite interesting was that we hypothesized that, you know, for doing cancer prognosis, like can this method elucidate tumor immune co-localization, you know, in driving predictions for favorable prognosis. So here across 9 out of the 14 cancer types, we found that tumor immune co-localization is statistically significantly increased in lower risk patients. So patients in the bottom fifth, so kind of all the patients in the blue. So kind of these are, so kind of this kind of corroborates with a lot of known literature on the prognostic information of the prognostic utility of TILs. And I think kind of where this finding contrasts with other findings in pathology, there's been a lot of like studies that have shown that, you know, tumor immune co-localization, when people segment these cell populations and quantify the co-localization and correlate it with survival, we do see higher correlation with patients with longer survival times. I think this work is slightly different, where kind of we aren't exactly giving the model like a priori where the tumor and immune cells are. You know, the model is just kind of ingesting in the entire whole slide, and it's almost like finding prognostic information on its own. And it just happens to kind of highly attend to tumor immune cells in the lower risk patients. So kind of this is just a potential, in training these multimodal networks, this is potentially how we could be using multimodal learning to kind of elucidate new novel image omic biomarkers. I guess that's kind of that's the first work that I've done. But I think, you know, over in doing this work in my PhD, I've learned a lot about kind of there's a lot of things that I've wanted to improve on, such as better multimodal fusion strategies, as well as learning context for survival. I'll briefly cover these very quickly. So kind of this study is called MCAT, for multimodal co-attention transformer. And kind of this study, we wanted to see like, can we do better than ensembling and or late fusion between histology and genomics? So, so in Porpoise, we were kind of doing this late fusion where we were just extracting, pooling all the patch features and simply just fusing it at the late stage of the network for doing survival and analysis. But I think one of the challenging things is that pathology and at least whole slide imaging and bulk transcriptomics operate at different scales. So it's not exactly spatially resolved, in which like it would be better to, if we had spatially resolved genomics, we could do both like early fusion of patches and genomic information early on in the network. So overall, this was asking, can we do better than late fusion? And I think also as to preface, a lot of my background in work in kind of before doing my PhD was in natural language processing. So a lot of my ideas have since been inspired by recent advancements in NLP. So kind of this is showing that how how multimodal fusion was done for audiovisual speech recognition, and kind of how we analogously did it for CPath. I think one of the more nuanced things that I wanted to, I thought weren't as could have been improved on was that, though in Porpoise we were finding, the relevant information in both histology and genomics, we're not seeing how these features are interacting. Like these features are interacting, kind of very, and like the feature space, but it's very hard to elucidate what are these interactions. So kind of the previous method was based on Kronecker product-based fusion, where we would take the Kronecker product of each unimodal feature vector. And I think kind of the method has been shown that so this method, also called bilinear pooling, is based heavily on a lot of advancements in audiovisual speech recognition and in VQA. Method works very, very well, and we kind of showed empirically that it does decently well for survival and analysis. But the main limitation is that exactly these interactions are opaque and difficult to understand, like how exactly pathology and genomics are interacting. So kind of so again, kind of my background is in NLP. I've been very fascinated by a lot of the advancements that's happening in NLP over the years. And I think one of the most recent, back then, one of the more recent advancements was in this ability to kind of do visual grounding of text and objects in the image. So this is an example of like how modern VQA techniques work, where if we have the text, what are these animals, and we have objects in the image, we can localize that the word animals localizes with the word giraffe, with the object giraffe in the image. So I think this is like very cool, showing how this is called visual grounding, where we can ground the objects in the image with text. I thought that this is this would be very fascinating for like if we can do the same thing with whole slide imaging and genomics. So the hypothesis of this work is that, can we learn co-attention interactions between histology and genomics, like word and image embeddings? And this is the method that we developed for kind of solving this task. And this is the overall method, high level view. We take, I'll go into this like one by one, but we take we take whole slides, we do similar patching of the whole slide and similar processing of genomic features. However, instead of just simply feeding it through a fully connected layer, what we did was we organized the gene features using the mutation signature database. We took these gene features and we organized it into gene sets. So using gene families, there's approximately 6 that we found. There's 8 gene sets, but 6 of them I think are redundant with one another. So what we did was we took these gene features, organized it into gene sets, and treated these gene sets as almost as embeddings. So each gene set is this like, let's say for tumor suppressor genes, it's a 1/82 gene signature embedding, and we would do this for all six of the gene signatures. And I think the key trick is that once we have both modalities formulated as set-based data structures, we can then compute the co-attention between these two modalities. I'll just to briefly walk through like kind of how this co-attention works. So let's track. So this is our collection of genomic signatures. These are our histology patches. And let's say that like this embedding here corresponds to oncogenesis. So what happens is that in the transformer of self-attention, we would, using the genomics as the queries and the histologies as the key and the value, we would compute the dot product similarity of how each histology patch is similar to this genomic signature embedding. So it will be around this row right here. So this is this is Q times K, and then we take the softmax of these scores, and it'll be 0 to 1, and then these scores are then used to update the values. So these scores are used to update the values, and this produces this matrix here. The genomics is the queries are left untouched, so this just returns the input. But overall, what happens is that for these patches, based on their similarity with oncogenesis, we learn attention weights, and these attention weights are used for aggregating all the patches to kind of form this slide embedding or summary that reflects the genomic signature. So visually, what this looks like if we visualize the attention weights is that for oncogenesis, a lot of the high attention patches that has dot product similarity with oncogenesis is in fact tumor patches from what we find. So this is just us saying that let's say if these are some visual concepts: tumor, stroma, lymphocytes, and necrosis. What we find is that a lot of the high attention patches that correlate with oncogenesis is tumor aggregates. Vice versa for the growth factor signature embedding, we see a lot of stroma. I think overall this co-attention is done for each of the six gene families. So the output of our co-attention is almost this like, whereas regular attention MIL pooling, whereas attention MIL pooling just takes the softmax of learns, whereas attention MIL based pooling just kind of learns one aggregation of all the patch features in the whole slide bag, with co-attention here, or what I call genomic guided co-attention. If you have K different gene families or gene signatures, will produce K different summaries of the whole slide that each kind of summarize a different portion of information in the tumor microenvironment. And I think I really like this kind of way for aggregating information, because of course in histology it's not like object centric. There's a lot of different visual concepts in the whole slide information. So kind of learning, this is kind of almost like kind of aggregating different parts of the intratumoral heterogeneity within the whole slide. And I think out of the box, this method performs quite well versus previous permutation invariant pooling methods and their kind of multimodal extensions with concatenation and bilinear pooling. These were on the same splits as the previous Porpoise paper, and we just chose the 5 largest cancer types to kind of to kind of to mitigate any overfitting. And overall, what I really like about this method is that kind of out of the box, it works quite well in demonstrating pretty good multimodal improvement, as well as I think having a better, more kind of, I'll say, interesting interpretability, where at least where now we can better localize, where we can better localize what are the important histological features that interact with all of the molecular features. So this is showing for a TCGA breast slide, kind of how all these different gene families light up different regions in the whole slide image. And I think that's okay. I got,
there's a few questions coming in, so maybe we'll start to field those. So first, if you can go to the previous slide on the results.
Oh,
so just linking it back.
Oh yeah.
Yeah. Okay, perfect. So just linking it back to your first work on Porpoise. So can you explain what strategy on the left is used in the Porpoise? And can you explain whether you think that using this strategy would further improve the results that you got in Porpoise?
This strategy, so Porpoise used attention MIL with bilinear pooling, but it used a few more. There's a few more hyperparameters in Porpoise. Like it used a lot of like gating. It used something called gating-based fusion, where one modality gates the importance of another modality. Here, the exploration of different multimodal strategies was just limited to just conventional concat and bilinear pooling. So this is where Porpoise is right here.
Okay, amazing. So I'm going to ask a couple more questions. So we've only got probably 5-10 minutes left. So if we can try and keep the answers nice and short, that would be amazing. So this was a previous question, but I thought we can ask this now because it's still on the topic of integrating different modalities. So Alex asked, so on the topic of incorporating genomic and pathology information from TMAs. So first of all, did you look into that? And also, well, yeah, so how would you go about that if that was the case?
I think for tissue microarray cores, from my understanding, they aren't often used in like current like clinical translation. However, except I think it'd be really interesting to, I think some diseases such as a lot of the blood cancers, lymphoma, they do use TMAs. So I think in terms of like as overall strategies for multimodal fusion, I think they would, but overall, I think whether it's for clinical purposes or for research, I think these strategies for multimodal fusion, like late base fusion, or what MCAT does with kind of the pseudo early base fusion, I think these would translate quite well for TMAs. Obviously, there's like less patches in TMAs, or like around like 4K by 4K pixels. So.
Okay, great. So David asked, can one locate genomic information within a whole sort of image, perhaps on an adjacent section?
Oh, I'm gonna read that again. Can one locate? Yes. So if kind of the way that this method works is that you just kind of within cotention, we kind of give it a large bag of histology patches. So if this bag has information across or have patches across multiple whole slide images, then we can localize genomic information not just in one slide, but across many sequential sections.
But I meant locating by using sort of biochemical means, not by using image analysis.
I see. I may not understanding the question. Could you repeat the?
Well,
if it depends, sorry, David, because of the time, if it's okay, you can reach out to Richard after the presentation. Just so I know Richard has another topic to also discuss, and we've only got 5 minutes left.
Yeah, fine.
Okay, great. Thank you. You can continue. Thank you, Richard.
Okay. I'll answer Fayez's question at the very end. I'll get to it soon. I think this work was like an early foray into, but I'll just like answer it now. This work was an early foray into doing early multimodal fusion. I think there's a lot of future work that we have planned to kind of look at, you know, once we have like basically for all of the going deeper into analysis, like what we did with Porpoise, where in these high tension patches across the different gene signatures, you know, look at the cell populations and then use tools such as HoverNet to kind of validate that, oh, you know, for oncogenesis, it is primarily tumor cells, and for cell diff, it's primarily, or cytokines, it's primarily immune cells. Also just showing that one of the limitations of this work is that we use a very narrow set of gene features, or we use a very narrow set of, or very actually broad set of gene sets. So kind of these gene sets do not reflect discrete and like more unique biological functional pathways. So I think there's a lot of interesting work in line where we're doing MCAT, but now with more fine-grained gene sets, such as hallmark pathways. There's actually one work already in ICLR 2023 that's doing exactly that, where they're using hallmark pathways and instead of using the gene families, they're using hallmark pathways and then doing an MCAT-like strategy for correlating it with his histology. I think overall, from the works that I've seen use MCAT, it looks like it does pretty well, and I think the method is overall intuitive to use. Okay, I'll just jump towards this kind of work that appeared in CVPR. I'll try and give this in 5 minutes. Overall, these are the challenges. These are the challenges for kind of weakly supervised learning in 4 whole slide images. You know, and I think the primary motivation of this work is that, you know, it was not so much on the multimodal side, but trying to solve this like problem in the red box, where how do we learn context, and maybe how do we broadly do self-supervised learning at the whole slide level, where we're not just trying to learn, you know, feature learn doing unsupervised learning at the patch level, but also we initialize the aggregation of all of these patches with kind of pre-trained features. How we kind of do that is that we kind of going back and looking at kind of the inductive biases of pathology. I think one unique thing is that even though it's a very complex computer vision domain, images are fixed at a given scale. So for 20x, the approximate resolution, the magnifications per pixel is 0.5 microns per pixel. So I think what this means is that I think broadly in terms of like future work in pathology, we perhaps don't need to learn things such as scale and variance because the scales don't change at a given magnification. I think 2 things is that I think pathology is a very unique object by part whole hierarchy, where, you know, these objects aren't like objects, or objects in pathology aren't object centric. It's almost this turtles all the way down approach, where, you know, we have cells that fit within kind of cell to cell interactions that also fit within broader kind of tissue interactions, and then the overall tumor microenvironment. So it's a very unique modality where like kind of each part is almost semantic, and it kind of fits within, so each part is an object that fits into a larger object. And kind of the analogy that I see here is that this is actually very similar to what's being, this is actually kind of breaking this down. This is essentially how long documents are processed in NLP, where this is almost like a word, a sentence, a paragraph, and then the document. And maybe just to jump on this year's comment, natural images will be closer to natural language too if we took a large enough image. maybe. I'll get into maybe. I need to think about that more. At least within pathology, I think it's much closer to natural language, because each of these parts, each of these visual, there's almost like, maybe for a pathologist, from people that I've talked to, there is this like visual vocabulary that's waiting to be tokenized and learned, where there's not that many, like there's almost everything, almost all the visual concepts, even if we haven't quantified them yet, are like known in the H&E image. Maybe I'll talk about that later, but it's a kind of there's this like objective. I think overall these tokens in pathology are objective. There is this vocabulary that we need to learn. Oh, two minutes. I'll just show this one animation where we do where we kind of do this hierarchical approach for processing whole slide images, where we break it down, where we first patch at 4K, patch each 4K image into 256 images, patch each 256 into 16 by 16 patches, bottom up aggregation via transformers. I think the key thing here is that we can almost do self-supervised learning. We show that we can do self-supervised learning for 4K patches. Then how this is done is that actually the main trick to kind of getting this all to be self-supervised is that, if we first do layer-wise self-supervised learning. We first do self-supervised learning at the 256 image level with 16 by 16 tokens. And the main trick here is that if we want to do self-supervised learning at larger image scales, all we have to do is use this encoder as the tokenizer for 256 patches here in this like kind of larger scale VIT. And the trick is that, even though this is a larger image resolution, the number of tokens is the same complexity as the previous 256 problem, where there's 256 16 by 16 tokens in the 256 size image, and there's 256 sized tokens in a 4K image. So these are the same complexities in the problem. So, so this is a strategy for doing self-supervised learning at scale this is a strategy for doing self-supervised learning at scale. I'll just jump into some of, most of this work is in the YouTube oral that we have, but this is just showing that, training using HIP, these are some of like the self-supervised T maps that we're able to generate, where we can localize tumor, stroma, immune information at the 256 level, and more broader tumor cellularity and tumor stroma interface at the 4K level. And I think I'll just leave it at there. But most of this work is within in our CVPR oral on YouTube. So please check it out. And also thank you again to all of my collaborators, and also my PI Faisal, for kind of providing this, for kind of creating the space to do awesome research.
Amazing. Thank you, Richard. Some really, really interesting work there. And not only was it interesting, but you explained it really, really well. So, hence the amount of questions. Obviously, everybody else found it really interesting as well. So I'm going to just ask two questions that I promised that I would field, and then we can leave it there. So one that was asked by Mohsin earlier on. He said, for different patients, survival time might be different. In TCGA, what is the longest and shortest survival time? And have you done any analysis, such as how's the algorithmic performance for different patient times?
I think longest and shortest. Longest might be like past 15. I think I've seen patients live as long as 15 years. I think shortest, definitely I've seen a lot of patients, there are some patients who only live up to who only live up to 4 to 6 months. There are some issues with, in terms of the ordering, I'll just jump to here. In terms of how the duration of different survival distributions affects affects survival. So the way that we've been training these and evaluating these survival methods is just via C index, which kind of looks at just the ordering of survival times, but not necessarily the relative the actual calibrated time distances, such as 1 month versus 10 versus 10 months. So I think for at least for training these methods with Cox losses, we're not really taking into account, we're not trying to create calibrated scores that would like rank patients, that we're not trying to get calibrated scores. Rather, all we're doing is trying to see whether or not we can order the predicted risk scores correctly in kind of the same order as the actual survival time. So it's not calibrated. And I think there's a lot of interesting work in using more using more metric-based survival times that do take into account the actual time distances.
Okay, amazing. So last question. So Fires asked, well, he first said brilliant work, and then he asked, given that the protein groupings, so this is on your work on MCAT. So given that the protein groupings are all related to cancer, it's not surprising that all of them would light up tumor regions. How did you validate that the image regions that light up for one protein group and not for another are actually associated with the gene groups?
Yeah, so Fayez, for this work, we had 2 board certified pathologists that like went in and introspected close from 50 to 100 kind of these co-attention heat maps in a similar way that Porpoise was done. So with Porpoise, we actually asked 2 board certified pathologists to go in and look at every single interpretability heat map across the 6,000 patients, across all 5 folds. We didn't do something at that scale for MCAT, but we did look through 50 to 100 cases. In the supplement of this work, there are some visualizations show that a lot of that for cancer types such as glioma, there's only around, like, even though there's 6 gene families, maybe 2 of the heat maps are unique. Or two are unique. For other cancer types like lung and breast, we do see more at least 3 to 4 unique co-attention heat maps.
Amazing. Okay, thanks again, Richard, for the amazing talk. Really well explained, and the concepts were really fascinating. So thank you. So this is my last seminar as chair. So next, I'll be handing over the seminar series to Adam Shepherd, who's on the call. So maybe, Adam, you can just introduce yourself for those that have joined online.
Yeah, sure. Thanks, Simon. So hi, everyone. For the people that aren't actually from the TIA Centre, so I'm a research fellow here, and I'm looking at precancerous lesions, precancerous oral lesions, that is. And yeah, I'm really excited to take over from this seminar, and we are hoping to try and keep it with the same sort of format that Simon's been doing. So keeping it to the 1st and 3rd Monday of the month. But I'll be sending out more emails before then. So, and also I'd like to say thanks to Simon. So I've seen a bit the background over the last 2 or 3 weeks of how much work you actually had to put into these seminar series. So you've done a great job.
Thanks, Adam. And
Yeah,
Richard, if you want to be added to the seminar series, then just give us a shout and we can make sure that you receive the invites.
Okay.
Great. Thanks, everyone. We'll have a great rest of your day, depending on what time it is. And I'll see you soon. Thank you.
Thank you. Bye.